Great.
Okay, you should see a browser window open right now. Is that correct?
Yep, looks good. Fly mine.
Okay, cool. So yeah, first of all, big thanks to our intern Roshni for coming up with these designs. So I think it made a big difference, and it also helped us implement many features we were missing. So I guess I'll start with the navbar. It's pretty much all there. Hopefully it looks cleaner, and yeah, otherwise it's not that different. There's also a logo now, which we thought looked very cool, and it was all Roshni's idea. I can scroll to the bottom to see the new footer. So the footer is slightly dependent on your mind. So I think you can specify your own blog. It will link to that there, and the same with the Twitter and your email. And the same with the citation. It will show your my name here instead, and the citation you set in the BlueGenes, in the web properties. And the funding, I think this applies to, it applies to BlueGenes. So the funding that you have specified will show here instead. So this here is configurable through JSON. So it's going to be in the web properties as well, but I don't think we have implemented that on InterMine yet. So now to the home page. If you have a mine description, it will say below the title, and here you have the same quick search, which is similar to the one in the top. Below here you have the templates. So these templates are automatically determined based on an algorithm involving the rank, which is how many times they've been run. I saw that in the JSP web app there was, you could configure it by setting some tag on the templates, I think. But when I looked at most of the other mines, very few had actually used them. So I thought this would be a better idea, but if you want to configure both, just let us know. And yeah, it sends you to the template page. And if you click on a specific one, it will open it like I expect. Here are just some large links to guide you to the correct page. So this sends you to the upload page. This actually runs a query to show you the data sources that are used for this mine. This is for the query builder. This is the user documentation, which will hopefully update for BlueGenes soon. The developer docs and the InterMine blog. Here we have a mine switcher that it has the same function as the one at the top, except it's supposed to be more explanatory to maybe those who are not familiar with the concept of having multiple mines. So it will show all of them, and you can read the description by changing them, or you can also filter them down to the neighborhoods, which I think are defined on the InterMine registry. And here are some external tools that will open in a new tab, like the data browser and Intermod Go. So hopefully there will be more of those that we can add as well to send people there. And there's a feedback like there was in the whole JSP web app. Yeah, that's it for the report. But I'm in the homepage. So yeah, just let me, if you have any quick questions, you can just come with them right away, or else I'll just continue on. So the list page is quite different and has much more features now. So you can filter by pretty much any text. You can search by description, name, type, if you have any tags, which I don't have currently. And there are fields, so you have pagination, and you can choose by default. So there aren't many lists on Alpha FlyMine, but it will by default limit to 20, but you can increase that. And you can filter by type, tags, or date, which goes from the last day to the last year. And also the type of list, which is private only, public only, and folders first. Because by default, folders will be spread among based on the data created. So by default, it will sort with the newest at the top. So I have a folder here, and it may be a bit confusing to begin with because it doesn't indent the contents. So instead, there is just a thick border until the folder ends. So this is a nested folder inside here. Hopefully it's not too confusing, but I don't think there will be nested folders very often.
Why don't you color
Yeah.
one of the bars when you open it so that for every open folder there becomes a sort of visual separator? I know you say it makes it bold, but if it was
Yeah.
colored, you know, just a faint blue or something, then it would be more obvious.
So like a different color for each folder, you think?
No,
Or just,
probably the same for each, but that line, which is my folder or my first
yeah.
list, if they were a different color, then that would visually break it up if you weren't indenting them.
Yeah, that's a good idea to use blue instead. I didn't think of that.
Or the mine color or something, you know, a pastel version of the mine color.
Okay, so moving on. There's an edit button now, and here you can add tags and so change the description, and you can also change the current location. So this is made to look like the directory view in any Explorer, file explorers. So you can create a new folder, but I don't want to nest it anymore, or you can use the arrow up to bring it up. So let's bring it to the top. Maybe create a new folder and then save it. And then, here we go. So yeah, we should have all of the features that that we have in the JSP web app. So we also have the combine, intersect, and the set operations. So that they stick to the top to make it much more easier. And when you select folders, you will get this bar so you can perform multiple operations on it if you want to move a bunch of lists into a folder or delete a bunch of lists. And when you select 2 or more, you will get the option to run a set operation. So there are still some fine-tuning that needs to be done on this page. So it's not final, like for example, the set operation symbols, they aren't correct. But hopefully the new model is clearer than the old one. So they're all pretty similar, except from the operation, except for subtract lists, since you can move the list between the groups that will be subtracted or not.
I really like that. That's a much better than before. And before, actually, the way the subtraction worked was very ambiguous.
Yeah, I'm glad we spent a lot of time on that, I remember. And Roshni did such a good job with the designs to make them clear and easy to understand. So now we can open a list and go on to the results page, which I regard as a work in progress. It's still largely the same. The difference is that now you can see some list details in the top. If you're opening a list and not a query that you've built on the query builder or running a template, then you'll get all the tags, the date created, count, and if there was a description, you'd get it here as well. And the enrichment column has been made clearer, hopefully, with the colors so you can see which one actually have contents. And yeah, so on the list analysis page, as it's called on the JSP web app, it still has much more features. So I think we'll discuss among the team which are important that we want to create before or add before releasing BlueGenes.
Just one quick thought. For any given user, they're probably likely to only want to use one of the languages. So does the drop down, you've got JavaScript at the top there, does the drop down, is it sticky? Does it remember what you used?
No, it's not sticky, but I remember to change it to Python, and I'll definitely do that. And to make it sticky will be more work, but if it's something someone really wants.
It's probably not that important, to be honest, but it'd be nice to have Python first.
Yeah, so that's on the to-do list. And we also have much more visualizations for the list analysis page now that haven't been added yet. So now moving on to the report page.
Oh, this is looking great.
Yeah, thank you. I'm glad you like it so far. So this is the new report page. So you have a summary, which shows all the attributes of the class you're looking at. In addition, it also shows the FASTA and the chromosome location. And if you click on that, it will jump to the region search and immediately run a search. And you can also download the FASTA. So I can start with the right sidebar here. It shows all the lists that this gene is a part of. If you hover over it, it will also show the description. And clicking it should run the query or show that list. Oops, I'm guessing it's the enrichment that's running now. So we will add the other mines here to show homologs. That's still a work in progress, and the same with external resources, which will show external links to Ensemble and other external sites for the same item you're looking at. Data sources is working now, and it runs a query to get the data sets for the specific class and shows them all here along with their external links, which will open in a new tab. So below here is the layout that I've created for alpha-Fly-Mine. So it separates everything into sections, and then it shows them in these collapsible IM tables. And on the left, there is also a table of contents. So you can click on specific ones to jump to them, or you can also click on the class or the item to go to the top. And these sections can also be expanded or collapsed. And you can also change in the layout editor, which I'll show you later, if you want things like visualizations to be collapsed by default, as they can be pretty heavy to load. And some of them can be pretty tall as well. So we have some plans to make it more readable by having perhaps a compact version of the IM tables that instead of showing this toolbar all the time, it will be expandable. So any feedback, or if anyone doesn't like that, then it will be great to let us know. But that's at least an area we plan to experiment, so it will look more readable. So I think that's everything to mention about the report page. So there's a new admin page now, which you have to be logged in as admin on the mine you're looking at to get access to. And then here we have the report page layout editor. So hopefully it's intuitive and the short description is good enough. And I can explain it promptly now. So you get to define a layout for all the classes that are present in the model of your mine. So and there's also a default layout, which I don't have anything on now. And that it basically takes every part of the default layout that you define that applies to the class you're looking at. I'll explain it a bit more later. But so this is the layout that I showed you earlier on the gene report page for Eve. So you can add any categories you want. And on each category, you can choose classes, templates, or visualizations that apply to this specific class, which is gene in this case. So I can show you how they're added like this, and then you click the plus beside, and here you'll have options to set if they will show or if they will be expanded or collapsed by default. And you also have buttons to change the ordering. And here is a tag looking thing that tells you what it is, which is, so CDS is a class of gene in the model, and this is a tool, and this is a class as well. In addition to class and tool, we can also have templates. So in the same way, I created all of these categories. So hopefully that made sense. And I can, so for the other classes, which I haven't created a layout for, like let's look at the protein. And they will have auto-generated layouts. which creates, in addition to the summary, it creates 3 categories, one for visualizations, all of them are always collapsed. One for data, which is everything in the model, which is probably too much for some large report pages or classes with a lot of references and collections like proteins. And then templates that are runnable, and these happen to don't have result for this protein at the bottom. So yeah, I can tell you more about the default option, but it's more like an experimental thing, as I'm not sure if it will actually fulfill its purpose. So its purpose is that there's a lot of classes in your model, right? And you don't want to you don't want to create a layout for every one of them. And maybe you don't like to auto-generate the report, which creates those 3 categories for visualizations, data, and templates. So that gives you the option of creating your default layout. So what you do is, need a reference to, since I don't remember all the classes in my head that I want for Gmodel. So the idea is to group relate data in your model under categories. So for Gmodel, we might have transcripts, intron, exon, alleles. And then if you open a gene page, it will show the gene model categories because it has all of this under its references or collections in its class in the model. But for a protein that might not have all of this, I think it only has allele, that it will show the gene model, but since these aren't applicable to the protein, I think, they won't appear. But since allele is applicable to a protein, it will show the category only with that. So that works to the category level as well. So if you have a category like, so a publication, I'm pretty sure has none of this. It will completely ignore the gene model category and instead just go on to the next category if you have multiple. Yeah, and I think I've gone through everything I want to do. So yeah, it's still under testing. Perhaps we'll do some user tests, and especially on the report page and the new report page layout. Yeah, I forgot to mention that it will always prefer the class-specific layout, even if you have a default layout defined. The default will only apply to the ones without a class-specific layout. So that's how the fallback works. It starts with the class-specific report, then the default, and then the auto-generated, which I showed you earlier. So yeah, feedback on the new report page or the layout editor will be greatly appreciated. And you can also play with this yourself. So in the HackMD, you will find the link to this page, and it's accessible anywhere. And you can also go to your own minds. And if you're an admin, you will be able to go to the same page and define a layout, but it won't be persisted. So I also noted that in the HackMD that you're able to use the layout editor for other minds, but since they don't have the web service to save it, which isn't released yet, it will only be available locally and not to other users except for your session. So any questions?
Really nice, Kevin.
Yeah, thank you.
I think it's so much easier to configure than the current web app.
Yeah, so I guess it was a good decision to not go for XML.
Yeah.
One question, when is the Docker image going to be updated with this new version?
Okay, so I update the Docker image, the latest tag, on every release, and this change, I think I'll release it maybe in mid-November or late November. You can create the Docker image yourself if you want to. because it's pretty stable from what I've seen so far. So under this documentation page, if you clone the BlueGenes projects, then under Docker here, you just need lining and dependency. You can get it on your package manager. Then you run line-over-jar and you can build one. And then you have a local container. So this is on a branch now called the feature new report results page, but I'll merge it into dev soon.
Is those old plugins that you have for BlueGenes, they're still working with this new version, or?
Yeah, they're still working. And we had a change to the tool API at some point, but we've updated all the existing ones. And we still have lots of new, as you can see, there weren't that many visualizations here on the gene report page, but we have much more now, and we'll be adding them soon after making some adjustments done.
Most interested in the cross-searching capabilities. I think Rachel's going to talk a little bit after, but that's more like on the back end, right? So the cross-search that
yeah, that's the federation, right?
Yeah, the cross-mine search is like a Node.js plugin that you can add to it. So
okay,
it's more on the front end, right? So
a cross-mine, so it's like a keyword search where you get results from all the mines.
Yeah, and so is this the cross-mine is already implemented on this new version?
No, I didn't know about that feature, but it's not too difficult to implement, so we could look into it.
And the BlueGenes, at one point you told me that BlueGenes would only access mines that have a HTTPS as the main protocol. Is because on this website that on this page that you showed, we can go warm mine, and warm mine is not HTTPS. So is there any?
So currently, if your BlueGenes instance is hosted with HTTPS, it will look at the protocol and will disallow any mines that don't use HTTPS. But if you use HTTP, which this apps.inmine.org uses, then it will allow all mines. But I think
okay. So the base protocol will be the one that you select BlueGenes to go for, and then it will basically filter out the ones that are not HTTPS when you are HTTPS, but then allow everything when they HTTP. Okay.
Yeah, that's right. And at some point we thought about disallowing HTTP, but just for security, since you're doing logins over it. But I think it could be up to the BlueGenes maintainer. I mean, the BlueGenes who is running the BlueGenes instance.
Thank you.
So, any more questions?
Yeah, so in the past, BlueGenes, it used to say it is alpha, beta, right? So now is it, you have released it, and what is it? Is it integrated into 4.2.0, or I'm wondering how to use it basically? Because I use snapshot 911 in when we do our mine, there's a variable, right?
Yeah.
In settings.gradle. Maybe that's too old.
The latest one is 0.10.0. And I think that also says on the GitHub repo. To answer your question about alpha and that, I guess it's officially still in alpha, but it's probably more beta now because we're approaching release. And we still have, on the GitHub, we have a production roadmap which shows everything we want to fix before we release it. But I think it should be pretty soon now.
Okay.
Since this was one of the major things we wanted to fix, which was the lack of content in those pages. So hopefully the report page now has the same features, or most of the features that the JSP web app has.
So for now, like say in InterMine, when I do the snapshot version, if I do 0.10.0, then I can play with this locally install?
Yeah, so 0.10.0 I released for, that was in June. I plan to release this as 0.11.0 in November, hopefully soon. And if you want, I could probably send an email to the dev, InterMine dev list.
Sure, yeah. No,
Yeah.
I was just curious. That's all.
At that point, then it will be easy to just to play with it.
Okay.
And also the InterMine version, which I haven't released yet because the new
Daniela, you're very loud.
and no, I just said that Kalpana, if she wants to use the version 0.11, she need also the new web services implemented in the InterMine.
Okay, yeah, that's right. Just to note, Daniela, your microphone is very loud. Sorry. Yeah, so what Daniela said, Kalpana, was that we can't, yes, so this latest feature, the report layout editor, it depends on a new web service that we haven't released yet. So once we release InterMine 5 or what we will release BlueGenes with, then it will be available. But until then, you can still play with it if you clone the GitHub repo and follow the building.md documentation on the GitHub repo, or you can just
we can
let us
even
know.
look at it here, right? bluegenes.app.intermine.org.
Yeah, or it's BlueGenes-staging.
Yeah, looks very good.
So any more questions before we move on to the model discussion? Yeah, thank you very much.
Okay, great. So so I shall take over from Kevin. So we've had some discussion, mostly started by Sam, on GitHub about making a core data model for expression data. So I doubt that we can fully decide on this in the sort of next half an hour, but this is really to get some discussion going on how much interest there is in this, how we should do it, how this model should look. So of course, it would have advantages for cross-mine searches and for sharing visualizations. We should probably discuss what sort of expression data we would want this to cover, whether it would just be RNA-seq and variants of RNA-seq, or if we want to be bolder and create core models for other things such as microarray data, if anybody has that sort of data. So this, of course, would be a big model change, so it would have to come with a release. It wouldn't be backwards compatible with previous InterMine versions. So Sam, do you want to talk? Sam came up with a core data model. Do you want to discuss your core data model? Shall I bring it up on the screen?
Yeah, sure, if you want. I mean, it really was mainly based on your human mind model. I don't really have any strong feelings about the details. I just wanted to get something that we could use to load our data. But yeah, sure, throw it up on your screen share.
Just trying to see if I can find it. Hang on a minute. It's there, but it's not coming up. Hang on.
And this is in production on the LIS mine, so if you want to actually see it in use.
Maybe if you could share that might be easier than me trying to find the
Sure.
the GitHub.
Yeah, let me just go to share screen here now. Let's see, desktop share. How are we doing? You got me?
Yeah.
All right, let's see here. So not all of the mines have expression data, but for example, BeanMine does. And so I'll just show you the model through the model browser. So at the top level, InterMine object, we have expression source. And that's just the thing that describes where you got this expression data. Oftentimes here in the States, we'll, and at LIS specifically, we'll go ahead and download the reads from NCBI sequence read archive, SRA. So we will do our own analysis of the reads for consistency, for internal consistency. So you've got the SRA accession there, description, and the name, just a name that shows when you look at it. I guess we can just take a look at the source itself. And we're generally dealing with gene atlases. So one thing that's kind of different in my other work at Carnegie, I'm always looking at differential expression experiments. And that's not really something I've had to implement on the LIS mines, where you've got, you want to look at the difference between treatments and that sort of thing. So I think that's sort of that's probably more on the visualization end, because obviously the samples get named with the treatment you've given them. But I haven't written any visualization code to show, the results of differential expression measurements. There's no p-values and that sort of thing. And that's a little bit higher level, but anyway, that'd be something to ponder. So these are just expression atlases. They're just atlases of expression of a particular plant, which means includes a line. This is a common bean, Negro Jamapa. And what we have then is a collection of samples. That's the interesting thing. The rest of it's just this metadata, the bioproject, the description, short name if you want one, the sequence read archive accession, and then origin is this kind of a way of keeping track of where things come from in larger scale buckets like SRA. So all of our expression data, the origin is SRA, sequence read archive at NCBI. And then the bioproject is, of course, an NCBI thing. And then I actually enter that into the, that's actually a class, because it's nice to just go ahead and see its details here, rather than just linking off to it. The data set's just the files that the data came from. Publication, of course, and then the samples, have an identifier. In this case, we just use the identifier that's used in the at NCBI. But then you want to have a name, and that kind of tells you a little bit more details, a description. Usually, kind of for plants, it's like how old the leaves were, or how they were watered and stuff like that. Stage 1 seeds. And then the biosample is another NCBI attribute that we bring in. And then there's also a number, which isn't shown here, just for the heat maps to put the samples in a particular order. So that's the expression source. And we'll look at the rest of so the expression samples are bioentities. Not too surprising, which means they're annotatable. And we get down to bioentity here, and we see expression sample. We've got 15 of them from this particular project. And so it's got a number, which is it's just used for, typically for putting like seeds together next to each other on the heat map, that sort of thing. It's just a number that's added. So I could show that. It'll look just like that previous thing, except it'll have the numbers. So if I order them by number as they are, coincidentally, it goes leaf, leaf, stem, shoot, flower, pod, 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 seed, 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 root, tip, root, young root. So I just did that myself when I loaded this data in. I added those numbers. And then finally, you have expression values. And those are, let's see, they're not sequence features. Where are they? Expression source. Where's expression value? Oh yeah, it's down here. Actually, I lied earlier. That's a very top level. Not sure, it's not even an InterMine object. Oh, that's interesting. This was a tip that I got from Daniela, that you can create a class that doesn't extend, what is it? Basically, it's a good class for storing things that aren't searchable. If we look at it's a good tip if you haven't seen this, so I'll show it to you. So it's not an let's see, it's not an interface. Whoops, sorry. You know how when we define classes, we almost always use the is interface equals true? It is not an interface. Is interface equals false. And so that means it's just a Java object. It's not an InterMine object. And that is actually good. Because it means you don't store a whole lot of stuff. You don't store anything in the you know, that serialized table, the InterMine object table, which gets really clunky if you've got, you know, millions of values that don't need to be searched. So that's why this is lying outside of InterMine object. If I close InterMine object, you'll see expression value is there. So that's just a little tip, or maybe it was Joe. Somebody gave me that tip. So an expression value has a unit and a value, but it references a feature. This is a little change we made recently because could be a transcript, could be a gene if you like to reference expression to genes, could be a transposable element, because some transposable elements express in some way, which I don't understand, but that's true. And so it references a feature and it references the sample from which it's taken. That's the deal there. So if you, you know, we can look at, I'm just using genes, but the idea here is that you make it more general. In principle, it should be transcripts, right? Oops, I didn't mean to do that. Show the units. We do everything in TPM. So the unit is TPM, and Sergio pointed out that that should go with the value rather than somewhere else. So we stuff it in there, and then so this will hopefully just deliver a bunch of results for the first page here, even though it's a query and a lot of stuff. So there we go. There's starting out with the first gene alphabetically, and here's the values, and that's pretty much the model. It's working pretty well. You know, if I go to this gene, there's my little bar chart that I've written. I'm still using Canvas Express. So you roll over these bars and you see what the sample is in detail, or just roll over the sample. And you can click on the sample if you want to look at the sample page. If you have a list of genes, you get a heat map on the list analysis page. So yeah, so that's pretty much it. I don't think I missed anything.
Great, that's nice, Sam. Yeah, so I think this model's nice. We could, our RNA-seq model is a little bit simpler than that, but we could easily get it. It would fit that perfectly fine. we tend to bundle sample and the expression values together at the minute, but we could separate those out quite easily to fit this model. So I guess the question is what everybody else thinks about this model, and whether they have data that actually fits this model, and whether they'd be prepared to change data to fit this model. It would certainly have advantages if we want to share some of these lovely visualizations, because I know we've got a lot of visualizations now for RNA-seq data, and Sam's obviously got some as well. So it would be really nice to be able to share these visualizations with other groups more easily. I don't think we could get it of we've got quite a mishmash of expression data in our FlyMine and HumanMine, so I think this would really only work for our RNA-seq data, because obviously microarray data is more complex. You have 2 samples, and we have a whole load of experimental data that goes with that as well. So are there any thoughts and opinions on this?
So EastMine has microarray data, and like you said, a ton of it. It's a lot of. So whatever the model change, it has to be compatible, I think, to be able to load microarray data as well.
Yeah, our micro data it's definitely more complex for this. It would take.
No, but the model, I think I used whatever was in the core, or I don't remember. I should check. But how much I deviated from the core model to load microarray data, but I could look at that. And I think the next RNA-seq and microarray, whatever the model we come up with, should be able to support both, I would think.
Yeah, I think so when I got into this business, I was working with microarray data and RNA-seq, and I never really felt like they differ that much. I mean, yeah, you have the log of intensity in microarray data, so you put it, you take the exponent of it, and you get a linear intensity. And in RNA-seq, you have counts, which is a linear intensity. But I wrote a big web application at Carnegie, and I stored microarray data and RNA-seq data in the same data model. I mean, you have some extra metadata, or you might have extra attributes, but I'm not really, I've always felt like this idea that microarray data and RNA-seq data are incompatible is incorrect. They're just 2 different ways of measuring expression. And if in the end, what you want to do is see expression, you should be able to cast the data into the same format.
Yeah.
Again,
I think you're probably right. I think it's just historically we've been adding these different expression data sets for so long that our models are actually very different for the 2 types. But there's no reason why we can't go right back to scratch and load it all different.
Right. Yeah, there's nothing in this model there's no place where the word RNA-seq appears. It's just called expression, and you give it units. So, I mean, the unit could be, rather than, TPM, it could be intensity,
Yeah. I mean, we have quite a lot of different, measurements anyway, TPM and RPKM and
exactly,
all the others.
Yeah.
Okay, good. So anyone else got strong opinions on this?
In the model that's been posted here, the nucleic expression score, sample, and value. But even for RNA-seq, there was some. I did load some of the data experimentally. We had some information about this developmental stage, or would that be a separate
that goes
method?
into the description of the sample.
Okay.
So if you look, I'll just click on a gene here.
Sam, I think that does raise the question of whether these things link to ontologies or not.
Exactly, that's what I was saying. Not free text as in description, but I think.
Oh yeah. Well, that's a good idea, right?
Yeah. Some of our ontologies.
Which ontology would that be?
Like for plants, we have the plant ontology, which kind of deals with anatomy for the Atlas type of experiment.
Oh yeah, for the samples, right? Yeah, and those are. Those should be, Come on.
Yeah, so I did load RNA-seq data for ZenMine, this frogmine thing, and I see experiments and antibodies, developmental stage, tissue,
Right.
all this type of information stored.
Yeah, and that's already automatically, here, let me show you, that's automatically included because expression sample extends bioentity, which means it's annotatable. So if you take a look at that, I'll pick a sample, we'll look at the samples and their name, and then we'll look down at, ew. I don't have ontology annotations loaded. Let me look at a different mine. That's a bug. You always find things that are missing. Should have ontology annotations loaded in there because I do that for like QTLs. Maybe I'm not doing that for expression, but I ought to be able to. We'll just see if I did it over here. Okay, I'm not loading it, but it does extend bioentity and annotatable, so I can associate an ontology already with these samples, multiple ontology terms. I have to put that on my to-do list.
Okay.
So Sam, another thing, that might be worth considering, you kind of alluded to the fact that we're not dealing with differential types of experiments yet, but
yeah,
when we do, we'll have to think about, replication and those sorts of experimental design issues.
Yeah, I expect that I would tend to say, let's store the higher level data, the stuff that comes out of HT-seq, but you certainly would have for a combined set of samples, you'd have an attribute which would be like, replications or reps, things like that. And I actually have a data model, like I said, for this other app I wrote, which is pretty darn similar to this InterMine model that is designed for differential expression. So I would probably, and that's worked pretty well, so I'd probably use that as a starting point if I were to dive into that. I have the data. I could actually put together a mine that is designed to display differential expression and then just sort of see how it goes, mucking around with the data model.
Should we be building this in now, do you think, if we're going to go to the effort of changing models?
I'm not sure it would, I think it would be on top of this.
Yeah, be adding to it.
Because ultimately, I mean, even in, so for instance, in that experiment that I was telling you about, I still have all of this data. You can just look at bar charts and plots. It does have reps, so you have, but each sample has a unique identifier. When you do 3 reps of seedlings with a given treatment, it's, usually _1, _2, _2, _3. So the heat map starts to look a little bigger. But it's kind of nice actually to see how the reps compare, as I'm sure you've seen it a million times. So the expression data model isn't any different. What you add then is the p-values and the log of the fold change and all that stuff that relate to those collections of replicates. But I think this model would be unchanged. It would just be an additional layer.
Yeah.
Because when you measure expression, you don't measure differential expression. You measure expression, and so that's what this model is storing.
Okay, brilliant. So I don't know, how do we proceed from here? Do we Daniela, can we create an InterMine maybe with the model changes, and we can load up our data and see if it works?
Actually, yeah, the only thing that we said is that because it will be very demanding for us, because we needed to update all the loaders. So the idea were to work on this stuff after the BlueGene release. So first work on the BlueGene release, and after that working on the model changes.
So I think that gives everyone time to think this through and look at their data and decide if it works for them, and come back to us with ideas, suggestions, or agreement or disagreement. How does that sound?
Yeah, I would recommend that people give it a shot. Here's the model, and I also sent it out in the email. And the GitHub issue is for discussing. You know, I've made some changes from stuff that's been posted up to that issue. So
yeah, so
you know, I'm happy to kind of keep track of that and tweak my model when people suggest changes.
Brilliant, that sounds good. So we can continue the discussion on the GitHub channel then. And once we have the BlueGenes release, we'll revisit this. We'll probably bring it up on another call, I would think, as well. So just to discuss again, and when people have time to think it through and look at it.
Yeah, and maybe in the meantime, I'll do a little experimental differential expression mine for I got a lot of Arabidopsis data, and just to kind of think about issues there, kind of see if anything hits me that would affect this core model.
That would be good.
So I'll have that be my homework assignment.
That'd be really useful. Okay, brilliant. Is there anything else on the agenda, Daniela?
No, I do not think so. Yeah, just a reminder, we will think it through during the next community call, the first of Thursday on February, I think. Yeah, but that's it.
Okie dokie, thanks everyone.
Thanks everyone.
Thank you. Goodbye.
Thanks.
Thank you.
Thanks for the BlueGenes demo. It's looking great.
It really
Thanks.
is.
Yeah, it does look really good.
Thank you, bye everyone.